It is Editing Abby, giving you an intro to point out the frustrating and exhausting fact that throughout the entire length of this video, I say Harvey Weinberg, not Hardy Weinberg. H-A-R-D-Y is how you spell Hardy Weinberg. I misspelt it in multiple places. It does say Harvey in the vocab words. I'm so sorry. It's been a quarantine. So that's a thing to be aware of. Yeah. Enjoy my face. Bye. Hello all. Because it was requested, because people really liked this format last time, I'm going to do it again this time so that we can see my cursor moving around and I can point at things in this file. So let's do it. I wanted to emphasize the vocab project situation. I know I have not graded your vocab projects. I am the worst, it's true. This is happening today and tomorrow. It should all be out by then. I'm going to be super lenient because I've just been so behind and swamped because life. But if you want feedback, tell me so in an email or in your email that you send a project. So if you want feedback, please say it in an email so that I can make sure that you get what you need. Another thing I wanted to emphasize is please tell me what you want, what you really want for review session. Do it in the discussion groups, or you can email it to me. I don't really care. My current plan right now is to just go over the practice exam at you in a video format, not unlike this one. But if you have specific questions you'd like to ask, or if you really want me to do, like, I don't know, a live YouTubing thing, or God, I don't want to offer Zune. Zune is scary. I'm not offering Zune. But I could play with live YouTubing. That's a thing I could hypothetically do. I'm also going to open a discussion group for everybody. So that everyone can talk amongst themselves and with me about, and see each other's questions and learn from each other for the review session. So I will be doing that as soon as I'm done recording this video to make sure that that's a thing that everyone can do. Yeah, so that's the plan. Let's talk Hardy Weinberg. So this is a terrifying equation because it's a quadratic equation, but like harder and more complicated and exhaustinger. I hate math. Remember how I hate math? I hate math so much. One of the nice things about Hardy Weinberg is that it makes sense. And I love this figure because the Hardy Weinberg is just a Punnett square. It's literally just squaring the two individuals that mated. So we've got these two heterozygotes, they mate. This is our p squared, this is our q squared, this is pq. So you can use this equation to figure things out if you want to. This one right here. But you can also use this system. You just look at the Punnett square and make sure it all works up together. So using p plus q equals 1 is important, frequently more important than using p squared plus 2pq plus q squared equals 1. Let's talk about this PEQ. There are 20 minions on a spaceship that crash landed into a distant planet. One of them was tall. When future explorers return 1,000,000 years later, they find 2,000 minions, this many of whom are tall. Tallness is recessive. And it has 2 alleles. First question, allele frequency of the recessive allele in the founding population. That is just 1 person out of 20 were TT. I know what to do. I am now going to put my head back in a different location. So, if 1 over 20 is lowercase tt, that's our double recessive. Q is recessive, P is dominant. So That 1 out of 20, last time I checked, was 0.05 equals Q squared. Okay? And that means that the square root of that, which is 0.22, equals Q. Whoa! And P plus Q equals 1. So P equals 1 minus Q, or 0.22, which equals dramatic pause while I do this, minus 1 equals 0.776 or 78. Because I can math, 0.78. However, the question was just the frequency of t. So this is the answer. You could have just stopped there. It's done. Moving on with our lives. Now we're going to do the exact same thing, but now we have 783 over 2,000 divided by 2 zeros. 0.39, which equals Q squared. Therefore, the square root of that, 0.63, equals our new Q. So, has the tallness phenotype been evolutionarily selected for? The short answer is yes. And you'd probably get like a point or a half a point for this, because we can just know at a glance that this is statistically different. But I would force you to do a chi-squared test, which you've done before, and I'm not going to do it again. However, you can also run this exact same type of scenario and test with this thing. It's the week 13 PEQ, which you have access to this question, and you also have access to the answer key. I'll show you where. So here we are at the home page. You scroll all the way down to the very, very bottom. Okay, not very, very bottom. Click on these things. This is the thing that you want to look at. This is your PEQ. Okay, moving right along. This terrible question really doesn't work. It works kind of, sorta, if you change it in this specific way. I did it. It was terrible. Don't do it. Just don't do it. You can just skip that. I'm so sorry that I exposed you to it. So let's talk about the assumptions of the Harvey-Weinberg equation. These are the big 5 assumptions. So let's say there was a novel allele, a novel mutation in our lactose intolerance gene. What would happen? We'd no longer be working with just P and Q, we'd also have R. We'd have a third allele, and then we would have to deal with that terrifying 3x3 grid that Kevin talked about in his last lecture. So go look at that, because that's what it would do, essentially. So let's talk about non-random mating. So if there's non-random mating, there are several different types of non-random mating. There's assortative, i.e. a sexual preference. One sex develops a sexy trait that attracts the other sex, and sometimes is actually quite in the way, like very long bird tails or obnoxiously bright-colored spider butts. All of these are examples of sexual assortative mating selection. And in these cases, we'd have alleles that are overrepresented, even though they are arguably evolutionarily deleterious. And there would be an active selection towards those. So we would see a higher proportion of that allele in the population than it would have been otherwise. The second is negative inbreeding, or a negative inbreeding quotient. In this circumstance, you have an extra pressure to not inbreed. So there's less random levels of inbreeding than you would expect. In a negative inbreeding scenario, you'll have a disproportionately lower rate of recessive homozygotes and more carriers of those recessive homozygotes. And you'll have more dominant alleles being expressed. The other option is the opposite, a positive inbreeding quotient, or enforced inbreeding. And in enforced inbreeding, you've got more than random levels of inbreeding, which results in a disproportionately large number of homozygotes in general. But in practice, it means that there's going to be a disproportionately larger number of recessive homozygotes and recessive homozygous phenotypes that might be deleterious. So don't inbreed. It's a bad idea. So there are 3 active natural selection pressure directions, essentially. There's directional, where we're selecting either for or against a specific trait, and that's going to shift things out of Harvey-Weinberg equilibrium to result in more of the more advantageous allele than one would expect given the number of homozygous phenotypes that you're working with. The other option is stabilizing. This happens in a codominant scenario or a polygenic scenario, where the homozygotes have more severe phenotypes than the heterozygotes. And then there's going to be selection pressure against those homozygotes and towards a more normal or standard thing. And the third is the opposite, where we have disruptive mating scenarios. This is a very common sexual selection situation where individuals prefer to find mates that are more similar to them phenotypically. And that can result in essentially the development of 2 subpopulations or even 2 new species. My favorite example is lactose intolerance. Again, I am lactose intolerant, and so is my partner. Because I am lactose intolerant, I am much more forgiving of the lactose intolerant phenotype of my partner, and vice versa. So it is advantageous for both of us to choose to be with someone who is homozygous for this recessive allele that we both have. So if we were to be able to procreate, our offspring would definitely also be lactose intolerant, and we'd create a whole lactose intolerant subpopulation. Okay, so genetic drift. Let's talk about genetic drift. There we go. Small population sizes mean that more sexually promiscuous gametes will have a disproportionate influence on the gene pool. And people who randomly die will have an impact on those ratios because each random death is a large percentage of the population and of the gene pool. And you will get this file as well, this filled out file. I'm going to give you this video in it. This video is a spectacular description of the different types of genetic drift that will help you understand the idea. Again, all you have to know is the term genetic drift and what it means, but you can hear about the founder effect or bottleneck effect, that sort of thing, through this video. And gene flow, which is the one that makes the most sense to most people. Gene flow is just people leaving and arriving from outside, i.e. migration. Ta-da! That's gene flow. And gene flow can cause any types of changes randomly, depending on where people came from. So those are the 5 things. So with that in mind, Attached to the email where you get this URL is a PDF file of that survey that I hope you took. If you haven't yet, do it. If I get a bunch more people, I'll send out the results again. And that will be entertaining for stuff to practice on for the final. It just gives you some real-life genetic ratios in a population that you can putz around with and think about. And there's a wide variety of different types of genes here. For instance, let's talk about these side by side so you can see. So let's talk about these side by side so you can see what I'm talking about. We only got 21 people, but that's okay. We're gonna work with what we get. For the tongue rolling allele, it's a very simple dominant phenotype, one gene, two alleles that we're aware of. The pinky one is the same way. This is the recessive phenotype. This is the dominant phenotype. Dimples are interesting. It's only partially inherited. There's a lot of environmental factors that affect dimples. It's also likely a polygenic trait. So what do you think? Can we run a Harvey-Weinberg question on these polygenic traits? Think about it. Pause. Take a moment. Can we? We can't. We don't know how many genes are involved, much less how many alleles. Harvey-Weinberg calculations are one gene with multiple alleles. So, though we cannot talk Harvey-Weinberg, we can talk population genetics on this. We can ask ourselves, has it been changing over time? Is it evenly distributed? Is there a selection pressure? Another one is relative toe length. Relative toe length is polygenic, but it has ethnic and regional phenotypes. For example, Ashkenazi Jews are statistically more likely to have these gigantic big toes. I'm going to show you my toe now. I'm sorry, I apologize for toes. But look at my toe. It has this giant big toe. That's different from standard. Most shoes are designed, entertainingly, for these toes or these toes, and this is actually a really uncomfortable way for shoes to be, for me. Anyways, that's my life. Moving on with our lives. Anyways, you can read this on your own. One thing that I did want to highlight is lactose intolerance in the Swiss population. So lactose intolerance in the Swiss population is an example of founder effect. Generally speaking, us cold white people don't tend to be lactose intolerant, but because the Swiss population was isolated for a really long time and continues to be relatively isolated and relatively small, there are a bunch of genetic quirks in that population, and one of them is that they're mostly over a third lactose intolerant, which is interesting. Having disproportionately long arms. I'm so happy that some people have disproportionately large arms. I was not expecting this high of a turnout of people with this phenotype. It's a relatively rare phenotype, but it does have a higher proportion of people have this that are from equatorial climates, people who have African or Middle Eastern descent. So I have Ashkenazi Jewish heritage. My ancestors spent a lot of time in southern Russia and interbred with people who were Middle Eastern. There are several different alleles and genes that cause it, but it's still fairly rare. So you can look at it as a single gene causing a dominant phenotype. I have this phenotype, my mother has this phenotype, my uncle has it, my grandfather has it, but not my grandmother. So we can trace this, which I find highly entertaining. If I talk too long, I've probably talked too long. So that's the feel of this. You can use these data to play around. And for each one, do you think, just glancing at it, is it in a Harvey-Weinberg equilibrium, or would it be if this population was closed? And if you don't think so, why not? Is there some sort of selective pressure going on? Is there an unusual amount of active drift happening? I really enjoy the story of Hardy and Weinberg. I think they're super charming as characters in the world. Just a bunch of really awkward nerdy guys trying to be nice to each other. It's a great story. Yeah, and here is a nice short vocab word list. My apologies for the late video. I will make a video for the review session. If you don't have any particular requests, I'm just going to do this sample final here. And I will do it at you, focusing on things that I suspect you will be struggling with. Ask me what you want me to focus on, and I'll do that. Okay, that is all. Y'all rock. I believe in you.